Hi folks, welcome to Module 2, Week 5. And this week we're going to be looking at aging on a cellular level and talking a bit more about some of the cellular theories that we would have discussed in that first module. So by the end of this presentation, we should have a good understanding of the biochemical mechanisms that are thought to alter the cell over time. We'll also describe the telomere theory in some more detail and talk about how that has been associated with aging. That's a really common underlying theory of aging. We'll also talk about the role of some of the following processes: the cell cycle, gerontogenes, cellular apoptosis, and then oxidative stress. And then we will pick up the oxidative stress conversation next week as we speak about the mitochondrial theory and how the mitochondria is thought to be involved in aging. So this is really kind of a two-part conversation that we will start this week and continue next week. Now, aging, we know, we've talked about this. We describe it as a multifactorial process. It's been thought to be influenced by a whole host of genetic factors, as well as environmental and lifestyle factors. And so in this week, we're really going to talk about how the biochemical and the genetic processes are involved in aging. So the basic underlying theory, the basic underlying idea of the cellular aging theories on a whole in general is that there are two things that could be associated with aging. One is that there's some instruction, there's some predetermined factors that are residing in our DNA that have a huge role to play in how we age. And the second huge idea here is that aging is really cellular damage over time. So whatever the cell is exposed to, whatever the organelles are exposed to, whatever DNA is exposed to in terms of environmental exposures, and whatever damage occurs, that is what brings about aging. So the two big ideas of cellular aging is that there's instructions about our aging process written in the DNA, and that damage or exposure from our environment has an effect on the cells, and that brings about aging. Now, looking at the schematic here on the right, we can see some of the various processes and cellular organelles and cellular components that are involved in cellular aging. So we've got gerontogenes. These are thought to be genes that are associated with giving instructions to how we age. We've got mitochondria, which we'll talk about next week. We've got signal transduction. Again, there's a dampening in some of our cells' ability to communicate, and that is thought to be a huge component in aging. And then the big one is really DNA damage and the inability to repair that DNA and any kind of spontaneous mutation that may occur. So these are some of the processes that are involved when we look at aging from a cellular or an organelle perspective. First off, let's talk about the cell cycle. That's a huge factor in talking about cell growth and regulation of replicating in the cell. So this is basically replicated by certain proteins. And so what the research is showing is that the instruction about how the cell cycle is regulated can be influenced by damage to the DNA over time. So basically the cell becomes less efficient at regulating its own cell cycle and at repairing its own DNA and so on. And so it grows uncontrollably. This can give us tumors or cancers or other forms of mutation. And so these are some of the components that are thought to contribute to aging. It can also just have other structural and functional consequences such as organ dysfunction outside of cancers or tumors. They can just be general dysfunction of organs such as the pancreas bringing about diabetes mellitus, such as the kidneys bringing about kidney function or kidney failure. So we really believe that all of this stems from the cell's inability to regulate its own replication and repair. This brings us to speaking about gerontogenes. So these are genes, again, that are thought to have some specific instruction to how the cell should function. Damage to these genes can bring about rapid aging. So these genes are on a lot of different chromosomes. They're found in various places throughout our genes, and they're thought to be virtual in that they can influence homeostasis and longevity without actually being selected for that role. So what that means is that a gene may not necessarily be ascribed to a specific function, but if there's any mutation or any damage or any accumulation of toxins or metabolites to that gene, then the implication would be aging or a decrease in longevity, or even just throwing homeostasis off balance. Some of these genes are also thought to be important to the cellular processes, such as transcription, translation of proteins from DNA, and so on, or just DNA repair in general. And when we see damage to these gerontogenes, we see rapid aging coming about. And so it's really kind of a coincidental association. So basically we see this rapid aging and then we see dysfunction in some of these genes. And so the theory is that the dysfunction of these genes, these gerontogenes, can then bring about cellular dysfunction and ultimately as a consequence, rapid aging. Some of the areas that we can look at to see examples of this are DNA. So DNA damage is definitely associated with aging. And so we call this a gerontogene for that reason. We've got the mitochondria and mitochondrial dysfunction. So the genes that are associated here can also be thought of as gerontogenes, in that if there's dysfunction with these genes, we can see mitochondrial dysfunction and therefore rapid aging. The telomere theory. So another really interesting common theory, which is a cellular theory of aging, has to do with our chromosomes and how they replicate as well. So basically, chromosomes have these terminal tips or these terminal ends that we call telomeres. They're sometimes referred to as the molecular clock of the cell. When we're born, they're nice and long. And then as we grow, as a cell replicates X number of times, the length of the telomere subsequently shortens with each of those cellular divisions. And so after a certain number of cellular divisions, we're really getting very close to the actual chromosome, the actual genetic material, which can lead to consequences in terms of slowing down our cell metabolism, in terms of slowing down the efficiency of the cellular function, and then that is thought to lead to aging as the cells begin to die off at a rapid phase. So basically the idea here is that as the cell divides, it loses that buffering part of the chromosome, the part of the chromosome that can eventually, that doesn't code for important genes or important genetic material. And so we can stand to lose that buffering part of the chromosome, but beyond that, we start to see some cellular dysfunction as a result of that. And so senescent or older cells will progressively see shorter and shorter and shorter telomeres. With each replication, with each cellular division, we lose about 100 base pairs from those telomere ends. So when we're a baby, we have a nice substantial length of base pairs contributing to the telomere. But as we age, we can see that by the time you're in your elderly adulthood, you can see that there's a very short amount of telomere left. And so that is thought to contribute to aging. Now, the length of the telomere can differ between species, one species to another, but this theory, this idea, this concept of shortening of the telomeres is something that's common throughout cellular division. The telomeres also have a few other important functions in terms of protecting the actual area of the chromosome that is coding for important genetic material. So it kind of protects that area. It also helps to organize the cellular nucleus by serving as an attaching point. So kind of just organizing the process of being able to divide those chromosomes and replicate them, as well as allowing the full length of DNA to be replicated completely. So it's just a buffering part of the chromosome where we can have errors, or we can have the actual important or vital chromosomes being protected within the bounds of those telomere tips. Another really important process we want to talk about is cellular apoptosis. This is basically cell death. This can be brought about to injured cells or cells that have determined that they're no longer functioning adequately, so they commit basically cellular suicide. This is programmed cell death, so this is basically a predetermined, instructed or prescribed cellular death that needs to happen for the overall benefit of the organ or the organ tissue. In order for cell apoptosis to occur, the cell has to shrink. It has to degrade or break down some of the important organelles, such as the mitochondria. It then kind of bubbles itself off into these blebs, as we can see here around the membrane, which is really just a part of it shrinking or contracting on itself. And it fragments some of the internal organelles as well. And then it eventually will apoptose or lyse itself. Let's move this, so that those tiny little blebs or apoptotic bodies, as we call them, can break off and just basically disintegrate themselves into cellular fragments. So that is how apoptosis occurs. Apoptosis is a crucial process to maintaining healthy cellular environments. If a healthy cell is in the environment of unhealthy cells, that can be detrimental to its own health. But we also need unhealthy cells to be able to regulate themselves and decide if I'm no longer functioning effectively, if my DNA is damaged, I need to go ahead and commit suicide for the overall health of the tissues and the organ. Apoptosis is actually a necessary process. We see this during fetal development. The finger web, so as we're developing in utero, we actually develop with these web-like structures for our hands. And so apoptosis takes place in between the spaces of our finger webs, and that is what creates the fingers. So apoptosis is not only a process that's crucial to cellular regulation, and to cells being able to destroy themselves if they're old or dying or they're damaged, but it's also a necessary process as a part of development as well. The final topic that we'll talk about here is oxidative stress. And so oxidative stress is basically the amount of oxidative byproducts and metabolites that accumulate as a result of some environmental toxins or environmental exposure. Things like the UV rays of the sun, things like some of the air that we consume has a lot of ROS or reactive oxygen species. These are thought to be free radical components that can be detrimental or damaging to our DNA. And so the accumulation of these harmful substances can lead to immune dysfunction, can lead to a decrease in wound repair or wound healing, dysfunction in the cardiovascular system, hearing impairment, and other sensory dysfunction, infertility, and eventually cancer or fatality. So there's a lot of harmful effects that can be brought about from the accumulation of these reactive oxygen species. And so to combat that, this is where antioxidants come in. And this is why antioxidants are thought to be so crucial to good aging and healthy aging, is that they offset the harmful effects of reactive oxygen species. The physical manifestations of accumulation of some of these RS components are damage to our DNA, oxidation of our fatty acids and lipids, so that can be influenced. Our ability to metabolize amino acids and proteins, that also can be influenced, as well as deactivation of enzymes. We know that enzymes need a specific environment to be functioning optimally. And so the presence of these oxidative components can influence how our enzymes and other proteins in our body function, including hormones, cell receptors, and so on. So they're just generally unhealthy metabolites, unhealthy substances that have a lot of implications for our proper, healthy cellular functioning. So let's look at some of the organs that are involved or some of the organs that are influenced by oxidative stress. We can see the skin, dermatitis, and other types of effects can be seen, for example, from UV rays from the sun. We can definitely get things like UV skin exposure. We can see joints being involved and leading to things like rheumatoid arthritis. In the lungs, we can see ARDS or asthma, which is thought to be involving oxidative components or being triggered by oxidative components. In terms of our vessels, there's atherosclerosis or vasospasm. So these are the overall consequences that are brought about by cells in these specific organs accumulating some of those oxidative metabolites that we talked about. Okay, and we're going to pick up this conversation next week as we talk about the mitochondria. The mitochondria is really the focal point of reactive oxygen species and RS damage. And so we'll talk a lot more about how the mitochondria is involved specifically as it applies to oxidative damage and oxidative stress. So now I want you to go ahead and reflect on some of these questions and also have a look at the articles that we presented this week. And then post your contribution in the weekly discussion board.